All right, it's been a while since we last met. Open Source Antibiotics Series 2, 10th of June, 2022. Today we're talking about the latest round of potency data that we got. And we were hoping to talk about mechanism of action stuff and tox stuff, and then paper writing. Those are the kind of main things. So I'm going to share screen because I think, yeah, Danny, I think you just posted the latest potency data, which is great. Thank you for that. All right, so hopefully you can see the screen. And all right, so just in terms of the most recent data that we got. So we had, just to remind everybody, we had previously had some fairly nice looking data on this compound 975, which had the para-cyano group. And it was a potent compound which had reasonable solubility and clearance data. Not perfect, but reasonable. And so we had made a set of compounds which varied that basic structure a little bit. So, you know, putting a nitrogen in the pendant ring in the northwest position, a couple of other substituents here, you know, other things in the ring to sort of zoom in on this motif as something we could play with. And we got data back from Paul Stapleton, who had assayed them all, and they all came back inactive, like red inactive, right? So we were a little bit surprised by that. And so we noticed that in that screen, there weren't any common compounds. So we hadn't asked Paul to put in one of the active control compounds. So we thought, okay, just to be sure about this, we would ask Paul to do a rescreen of a set of these compounds along with a positive control, both ours, so a compound that we tested before, and one of his, so an internal control. And so he kindly agreed to do that and came back with the data just a couple of hours ago. And the data were the same. So he again found that almost all of these compounds were active. There's a sniff of activity with this guy, but not much. So that looks like the SAR around this very nice compound with the cyano group, the SAR is very tight around that position. I guess we had an idea about this before. We have played previously with para substituents productively. And I think we tried some meta substituents and had inactivity. But it is pretty sensitive in the sense that now we're including a meta substituent alongside the cyano group, and the compound goes inactive. So the take home there is that we can't vary this compound very much in that region, and it aligns with previous data we've had, but it's a little bit surprising that it's so sensitive, I suppose. So the current plan, really, I suppose, is to fold in these data into the main body of the SAR, and and by doing that, to fold the data in to the paper in the same way. In the current draft of the paper, which I'll get to in a second, I've just added in a comment about the fact that because this top left-hand compound with the cyano group on it had the best balance of properties, we synthesized a little focus library around that structure and found them all to be inactive. But I think it's probably just a better route to go the non-chronological way of doing things and say that, hey, you know, the SAR is really tight around this compound, but it remains the one with the best balance of properties so far. So that's where we're up to on the potency side of things with the last compounds that we had. Anyone want to chip in with anything here? Suggestions, comments? I think it's pretty cut and dried, really. Okay, so that's where we're up to. Obviously, you know, it would be nice to include some of these. We don't have to include all of them in the paper, you know, necessarily. But we obviously, I guess this is on you, Daniel. We need to make sure we've got the data for the compounds, right? So they're for the characterization data for chemistry, because we're going to want to include them in the paper, even if just in the supporting information, and we can include a subset of these maybe in the actual paper, but we'll get to that. All right, just on the tox side of things. So I'm just going to stop sharing a second because I just got an email. Last thing you want to do is put your email on a public meeting. So I just got an email from Alex, who said that the latest experiment on binding didn't go terribly well. She's still working on the DNA binding experiment. So if you remember previously, Alex was showing some DNA binding stuff. I forget the nature of the experiment that she was going to do. From memory, I think it was that she'd done a one-way temperature change, and we were going to do that, but also in reverse, just to check that the reversibility of things, as far as I remember. The suggestion from the data was that there was no DNA binding going on, but I think she's trying to iron that out so that it's publishable. And I think that she is needing to order something and repeat an experiment. The tox stuff, she is also getting something ready to publish, but isn't quite ready to do that yet. But she is going to set up the DNA binding experiment for next week and try and finalize things at ASAP. Obviously, the paper is getting to a point of being nearly ready, so we're going to have to get that last data from Alex. And then on the, I guess we haven't got Lee with us today, which is a shame. So we've been in touch with Lee about the mechanism of action data. Let me just pull up something I got from him last. One moment. So I think that we, yeah, so as people may remember, Lee had done a screen on hex cells. I think it was mammalian cells to the latest MIBS run to find out some targets from mammalian cells. Had come up with a few hits that were presented at the last meeting. They were repeating that and doing the MRSA run. So we had a bunch of targets being suggested that were mammalian targets, and including, interestingly, this ALK5 protease, which came up. The origin of these compounds is that they were designed to hit that kinase, so that kind of was an internal control that suggested that the experiment was working, which is good. But Lee is repeating that and is also doing the experiment with MRSA itself. The idea really is to sort of try and find a target in MRSA and to try and explain perhaps why we're seeing mammalian cell toxicity with these compounds by identifying a mammalian target. So Lee is still doing those. He was hoping to come today, but I guess we may have missed him. So I can follow up with him on that. But I think he's got everything he needs to finalize those for the paper. As far as, so we took the hits that Lee had identified, and Daniel was looking at, the most similar compounds in the literature that have been found to hit certain targets. The ALK5 one, the original target for these compounds, came up, and there were a few others that I guess, Daniel, you were looking at. We're going to want to try and summarize those in the paper in a very succinct way, but I guess we can finalize that section when Lee comes with the latest data. Yeah. Okay, and then the other thing that we were going to talk about was, yeah, I guess just the paper. That was the next thing, really, for me. So I've taken a look at, let me share it. It's going to be easier.
Matt, sorry,
Yeah.
just before you share,
Yeah.
Maria actually does have a brief update in terms of chemistry that we've got going on. And that might also feed into kind of this question about how we talk about the nitrile, because she's been working on a few different kind of scaffold top kind of the nitrogen scan. I'm going to stop talking and let Maria talk.
Yeah.
Go ahead. You should be able to share.
Yeah. Can you see my presentation?
Not yet, but it might come.
Now?
Not yet.
No. Okay, maybe now, yes.
Yeah.
Okay, so we have few compounds ready or almost ready. This is ready to be shipped, and these other are under purification, so will be ready in a while. And we have these three compounds that the synthesis is in progress. In particular, for this final compound, the bromination is in progress. As regards the imidazole derivative, I'm having some problems in the last Suzuki coupling. And because when I use the imidazole, I'll try to put the, when I try the Suzuki without the a protecting group here, the Suzuki doesn't work. I tried different conditions, but I have just starting material and nothing as desired compounds. So what I'm trying now is to protect this nitrogen with this protecting group. And I'm trying again the Suzuki, but what I'm thinking is that these two nitrogen maybe can chelate the ligand of the Suzuki, and the reaction doesn't work for this reason. So what I'm thinking is that if this Suzuki doesn't work, I can change the order of the arylation of the imidazole. So first put this with the cyano, the paracyano, and as a second group, the pyridine. So maybe it would be better. I can try.
Maria, did you try microwave? Because in my case, this reaction just worked with under microwave conditions. Some of my reaction 4 hours microwave conditions.
4 hours. I tried the normal condition first and then microwave just 1 hour, but I didn't see anything. So just starting material.
Wow.
Okay.
You ran for 4 hours at the microwave?
Yeah, I did 4 hours microwave because I was following up by LCMS. I could see full conversion after 4 hours.
Okay, I can try because I didn't see anything, neither a tiny peak of desired compounds, so nothing. So I will try. And another
Daniel, Maria, sorry, Daniel, would you mind sharing the conditions that you were using? Maybe we need to change our conditions.
Yeah, we can share. I can share with her. I can share my ELN.
Thank you. And for the last compound, Bushi is having some problems in obtaining this intermediate because he doesn't see any desired mass on LCMS. And what is he seeing is the mass of this intermediate, but the identity of this intermediate is not sure because the alkylation could be on this nitrogen or on this other nitrogen. And maybe the cyclization doesn't come because it is on this other. So he's working on the isolation and characterization of this compound. And if this is the right intermediate, he wants to try the cyclization with this condition. But otherwise, maybe we have to prioritize this compound and wait for the result for the other compounds and then back again on this if it's
Maria,
yeah.
I have done this reaction a couple of times. I would try DNF and high temperature because your case here, I guess the first step is happening, but the second step, which is the losing the water to close the cyclization, you need to have higher temperatures. Need to be high.
Yeah, so I think, Daniel, we also need to understand whether this is actually the right structure that's on the slide. Mm-hmm. It's what has been proposed, but we're trying to confirm it by NMR before we really. So the idea is we're getting clean conversion to whatever this material is. We think it's what's on the slide. If it is, we'll isolate and then we'll push the cyclization separately.
I see.
If it's not, and the alkylation's actually gone on the other nitrogen, then it's never going to cyclize. And yeah, then the question actually becomes, how desperately do we want this compound? Mm-hmm. And so I'd be interested to kind of hear everyone's thoughts on, how hard should we push for this one? How hard should we push, basically?
I mean, this is the thing, they're all interesting. And it depends how much time you have. These will be great to have, but don't break yourself getting it. They all have interesting variants, no question about it. And it's good that they just have the cyano group because it looks like that's what's needed. I mean, I'd agree with Daniel in the sense you probably want to stew this up because all of this should be reversible. The reaction you're trying to do is a dehydration reaction, so you could try and get rid of the water as well to try and push it. So the DMF and heating thing might do something useful. But yeah, tough one. I'll keep us posted. I guess the question is the kind of time frame that you have here. You know, I mean, we, as we'll talk about in a second, you know, we're trying to finish the paper. We have until the paper is submittable to get all this stuff done. I agree these compounds will be attractive to get you know, evaluated and included in the paper. No question about it. So, you know, there's still time because we haven't got a paper in a submittable form yet. But we're keen to get something published for all of us. So I guess just keep us posted. You know, the sooner we can have these things evaluated, the better. Do you have a sense of a target time frame for finishing the compounds? I know it's very difficult to say, but Maria, do you have a sort of?
I think. Sorry, I was going to say for the compounds that we were just talking about. I mean the nitrogen scan; those ones should be ready in the next week. The one where we're obviously having this synthetic issue, that one pending confirmation of the structure of that intermediate. Yeah, that one I have no idea on at the moment. If it is the right intermediate and then all we've got to do is cyclize, hopefully it won't be too far behind.
Okay.
Maria, the imidazole, I guess, I mean, pending Daniel's conditions, perhaps, maybe we could get that one out the door pretty quickly also.
Yeah, I will know if the synthesis is working today. If this is not working, I can change the way of synthesis, and if Daniel share with me his condition, I can try also that. So I think that in a week I can know if I have the compound or not.
Could you go back a slide?
This one?
Yeah, all right. So those are all right. So the 7573 we haven't evaluated yet, right? That's new. Maria, the 7573 you haven't sent that to us yet, right?
No. This is ready, but we haven't shipped. Yeah.
Okay, and then under purification, there's a strong suggestion that those will be good to go. I guess, and then the reaction ongoing. Yeah, all right. So the in progress ones were the ones you had on the folder.
Yeah, the ones on which we have some issues. Yeah.
Right. So you're kind of, I mean, you're pretty confident about those top 4, and then depending on how things go. Right. Okay, great. That's really good. Well, I mean, yeah, based on the latest results, there's a compounds that certainly are relevant. Great, thank you. All right, let me show. So yeah, I was looking at the SAR for this paper, and I've sort of cleaned up the intro a little bit. And I was just going to walk through a few things that came up as I was looking through it. We were talking about this before. This initial figure is a little bit, as it stands, is a little bit unnecessary. It's useful to have the areas of the molecule highlighted, but to make it into the paper, I think it needs extra information here. So I think we need to just adapt it such that it has information that we had at the start of the project. So to describe basically this compound and what we knew from the initial screen that was done before we started. So things to do with, you know, a mention about tox, a mention about suboptimal clearance, a mention of the fact that there was potency against MRSA, but nothing else. Those kinds of things about the sort of day one fact sheet about what this compound is. And if we put that on the side there, then we describe the hit better, then the diagram is kind of more useful, I think. we need the structure there at the beginning of the paper to help the reader, but we need the diagram to be a little bit more interesting, I think. And then we were talking earlier about the synthetic schemes. So I think these are fine, but they need to be merged. So the scheme 1 and scheme X need to be made consistent and merged, I think, so that it's one synthetic scheme that gets to all of our different motifs here. So this one at the bottom, I know Daniel, you just put it in here as a placeholder, really, but I guess we want to tweak it so that it's like the others. It deals with the homologation thing, which is useful. But we need to make it generic like these. At the moment, we have sample yields as these XX%. And we can do that if people find that helpful. We would then need to have the ranges. And that means looking through all of the examples of these things that we've done to pick out ranges of yields. If we really want to do that, I'm completely agnostic about it. I don't find them terribly useful for a medchem paper, but if people would like to have them, then we're going to have to dig those out and give those ranges, which means looking at every single example of all the reactions. Any preferences? Say again, Ed, sorry?
It will be in SI?
It's all in the SI. Yeah, all the individual yields are in the SI. Yeah. And so if we want to have the average, not the averages, the ranges here, then we just have to go in and pick them out. That's all. What you want to convey is sort of an average, right? I mean, essentially what the reader wants to know if any of these reactions are nightmares. That's really what they want. But to do that, you have to provide the ranges. Yeah. Okay. And then what was this? Let me just hang on a second. I've got all your pretty faces in the way of exactly where I need on the screen. Okay. So yeah, that was just, okay, that's just a synthetic thing. And then the structure activity relationships. All right, so these tables are in the wiki, right? And were the tables that Dana pulled together very nicely for the wiki. And there are a couple of things, there are a couple of issues with these tables. One is that the VRE column, I think, is not needed, as people have said already. So imagine that we remove that and put that information in the supporting information, because it's not a main focus for us. So then you remove that column. And then what you have is essentially a tables linking structures with MICs, which is fine, except that you could imagine just having a chem drawer where you color code these things. So you have, red structures for 32 and, orange structures for 16 and green for 4, things like that, which would convey the same information, but in a simpler way. So you could imagine redoing these where you cluster similarity by doing that, or we just stick with it as it is. I don't really mind which way this is. All the tables need to be updated anyway if we're going to include the more recent data, but we have this choice about whether we tabulate or whether we chem-drawify. The advantage of chem-drawifying it is that you can put related structures next to each other in clusters, and you don't have blanks, which is what we currently have in the tables. Does anyone have a massive personal preference about which way to present the data?
I think I prefer a table. I guess maybe I'm also not sure what a ChemDraw will look like in terms of the way you're describing it. I mean, we could potentially try it. I understand that I'm now asking someone to put together a ChemDraw in this fashion. But I mean, we could look at it and see. In terms of clustering similarities, so I mean, we can just reorganize the table so that it's in, things are clustered together in a way that is, I don't know.
Yeah. So I mean, one way of doing this, provided there is similarity, and sometimes we get unstuck because there are fluorines different or a methyl different, and there are multiple differences between compounds. But provided there's similarity, you can sort of cluster things quite neatly in a kind of fan diagram where the compounds that are potent are green and close to the structure, and the compounds that are orange are a bit further away, and you just draw the structure and color them, and then the red ones are on the outside. And so you have a sort of core in the middle, and then you have these structures fanning out. It can be a nice way of presenting it. Ed did that for a paper we had on bioisosteres for a malaria project, and it was a neat way of conveying the information. However, in later, this is a neat table, the one that's on the screen, but for later tables, you start to get, so here, for example, you start to get slightly more differences between things, and it's not so easy to convey that on one diagram. I guess we could mock up one, see how it goes. We also have this issue as well, I guess, before worrying about the final format of the tables, we do have this issue about whether we want all of the compounds in the paper, of course. Some of these can be relegated to supporting information. When doing that, you do reduce the impact a little bit, I think, because it's nice to see just the variety of inactives here about how much has been explored. It always feels a little bit shortchanging if you have it in a medchem paper where you have a sort of, roughly equal number of actives and inactives, and you look at the supporting information, and there are a bunch of inactives sort of stuck there because those are useful data points. Perhaps what we need to do here is to make sure that all of the compounds that we have potency data for are included in these tables in the first place. And then we can make our final cut. So I've assigned, I think, Daniel, I've assigned you that task at the end of making sure that in the master list, every compound that we have potency for is in the paper. And if it's not, that we list the compounds that are currently not in the paper. I just want to make sure we've got every data point and we haven't missed anything. And the reason I'm asking that is because some of these diagrams from the wiki are a little bit older than we might remember, because Danny did these months ago, and yet we've got new data since then. I just want to make sure we've got everything. So that's the nature of some of those comments there. And then this is just more of the same. Yeah, and then there's some reordering down here. We make a very neat point in this Southwest substituent about the importance of the 2-pyridyl group, and I want to make sure that is clearly done here. And then later we can deal with the issue of the bridging nitrogen. So just to separate those out, and the comments are about that. One of the neatest bits of the SAR is where we demonstrate the 2-pyridyl structure as that is needed, that a 2 coordinating group isn't any good. It's a 2-pyridyl that's necessary, and I want to make sure that is really clearly done before we worry about the homologous series and the N bridging series. So the comments there are to do with that, and I think again that this has been assigned to people. Just going from the SAR to the ADME and physico-chem properties and stuff. There is a nice initial section here where we talk about the data we had at the start, and then the metabolite identification, and then of course we have all of this other data that we got, and there are 3 sets of data. There's this set on screen. There's a more recent set, and then not shown here is the stuff that's on the wiki that came from you guys at Northeastern. And we're going to have to combine them, obviously. And we're going to have to include some compounds in the paper and then put the rest in the supporting information. The data will be in the master list presentation, but we can also, if we want to, provide the data and the supporting information in the way that's shown here, where it's all sort of laid out. But I'm pretty sure that the best way of presenting a subset of the data in the paper will be to have this tabulated, because it's 5 or 6 data points, so there'll be columns in a table, but we'll need the structures there to get the most out of that. So we're going to have a table with structure and then column entries, and to make sure that fits anywhere in the paper in a reasonable way, we're going to have to limit ourselves to a smaller subset of these compounds. Before we do that selection, I've made some general points here in this paragraph that I wrote about the data that we had thus far. There are, I think, 3 or 4 GitHub issues where we discussed the data, where we had the data and had a meeting and then discussed the data. What I'd like people to do is look at those 4, I think it is 4 discussions on GitHub, and I've made a link of where those are. So down here. just to make sure that the points we've made there, if there are points made in those discussions that people think are important, that we paste them into the paper and make those points and therefore include those compounds in the paper. And for any compound that we don't mention explicitly, that then becomes a compound that can just be put in the supporting information. That seems to me to be a way of making that cut. If the compound is mentioned, we put it in. If it's not, it's SI. So if people could look at issues 80, 81, 82, and 104, as I've shown here, and just think about which things we haven't mentioned so far that people think should be mentioned. From my perspective, I've mentioned everything in this little paragraph here that I think is important, but people have different views about what's important and what isn't. So please do look through that and add things in as you see fit. All right, and then the last section here is on the tox, which we need the data on, and the MOA. For the activity against other parasites, we're going to be cutting that out, right? But we just need some holding pattern mention of the fact that many of these compounds were tested against something else and have shown promising data, which will be described in a separate publication. I believe that's how we're going to phrase it, right?
I'd like to do it like that, I think. Yeah.
No, I mean, no worries at all.
That makes the most sense.
Yeah.
Yeah, I agree. It's better to have a second publication because there are too many things to discuss. Solubility or cytotoxicity and potency. There are many compounds that are really potent against the parasites. So I guess a second publication would be better.
For sure. It just doesn't fit, which is fine.
Could be a short communication, I don't know, a letter, paper.
Let's do it.
Or full paper, let's see.
For the, so the references are looking okay. And then I've got a supporting information list at the end. I wasn't sure if this was there, but as I was going through it, I made a list of bits of SI that are going to be needed. We're going to need this anyway for the paper, but please do look at that. And if something is mentioned in the paper and is not there, then please add it to the list of things that we're going to have to add in. Okay, and then that was it. All right, any comments on the paper? Because it would be great if people could go back in and look at this document and try and make changes that are needed so that we can clear up all the comments on the right. Yeah.
I guess the first thing should be like organize the full list of compounds because I guess some compounds are missing here. And we need. I will check it this next week's how many compounds we have and how many compounds we have in the paper just to see if we are missing something.
Yep. It's absolutely necessary. So yeah, that's your big task, Ed.
Yeah, I saw that.
I mean, we just have to do that tracking of just making sure we haven't missed anything. That's all. And with a project like this that's inevitably a bit sprawling, we just have to spend the time to make sure we haven't missed something. There will, of course, be, the master list contains a bunch of compounds. We just really here want the list of things that have been evaluated and to make sure we haven't missed a compound. All right. Great. That was everything that I had, I think. Yeah, I think that's everything. All right. Any other comments anyone wants to make? Anything that's needed, I haven't discussed?
I guess just a quick question. Where are we hoping to submit this and what sort of time frame are we looking at?
Well, I mean, I think that there's nothing, the things we're waiting for are, Alex's data on tox and potential DNA binding, making sure that we have the MOA data. We just need to be really ready to go when we get all the data in. And of course, we'd love to include your latest compounds that we were just talking about. so that I'm keen to make sure that the paper is in as good shape as possible for when the data comes in and we can just fold that in. To my mind, this is a kind of ACS MedChem-let kind of paper, right? Something of that kind. ACS infectious diseases, that kind of thing.
I was going to say, it feels a little bit long to be a letters at the moment. I wonder if it could be a JMC with all of the additional biological data that's going in there as well.
Yeah, sure.
But I guess one of the other questions I was going to ask was maybe what we do from a Northeastern perspective is we put a time frame on this and say, you know, whatever's not ready in the next 2 weeks, we kind of pause on and just send what we've got. And then, you know, if there's a killer compound in the set that we have, maybe we reevaluate, but otherwise we kind of call it done at that point.
Yeah.
And then that way we know that we can get that data in. Or maybe the better way is to actually reach out to Paul and see if, sorry for the background noise, maybe we reach out to Paul and see when he would be able to run another assay.
Yeah, I mean, booking him in is the key, really. Yeah, so he has advance notice. But yeah, I mean, I was hoping if people could look at the paper in the next week and try and deal with some of these comments. Daniel, I know you're getting data together. You're going to give a talk on a little bit of this project at a Farm Alliance thing, which is going to be great, but it'll be a high level thing. But I guess you're looking at data for that. If you can deal with some of these issues in that kind of time frame, then we can get things in good shape. After this meeting, I'll go back to Alex and to Lee and say we met and we're keen to get things submitted ASAP. So if you guys were able to submit compounds in the next couple of weeks, yeah, then inevitably that would be then, you know, data back, I don't know, a week or two after that. And really that would be the end of the SAR. So if we could submit this in a month, that would be really great. That kind of time frame. Something of that kind.
Okay, sounds good. We'll push for the next couple of weeks, see what we can get done, and then we'll keep you posted.
All right.
The other question is, do we need LCMS traces, images of the LC component for the supporting information? I don't think that we've given you that, but if we do need to get that, we should probably get on that now if we want to submit within a month.
All right. So the ideal is, I mean, two things. One is we share as much data as we can, right? So if we have NMR data, we share the usual data, but we also share the raw data if we can, right? So this is an open science publication. We're meant to be sharing raw data. So the usual data, it helps if we have that too, if it's available. For the
so
LC
what would
data.
You need for that?
Yeah, I mean, so
like
Ed and Daniel.
File?
It's basically the files that can be used by, you know, Mastronova and things like that. Ed, Daniel, you've got data available in certain file format you can advise?
Yeah, I have.
Okay. So it's a, you know, whatever, so you take it from the instrument and you save it as a what?
Yes, and then I upload in the ELN.
What's the file format?
It's
Sorry, Ed?
Just have it as a zip file.
Yeah. Sure. I mean, it's just it needs to be a file that can be, you know, processed by for example, Mastronova, right? So
yeah,
that you can interrogate the raw data.
So if it's a compressed file, you can just zip it and then open it.
All right, so we can be in touch with anyone who's going to be helping to collate all that together, but it really helps if we can do that. But on the LC thing, so that's about demonstration of purity. If we have LC traces taken, then we should use those. If we don't have them, then to demonstrate purity, usually you just provide the proton NMR, right, and say that by NMR it's 95% pure. I guess I'm keen to make sure that if we have evaluated a compound biologically, that we have some way of showing purity. And as we all know, JMC will ask for that.
I normally share the MNOVA file because it's like the file that I solve it like with the chemical scaffold, and then it's the solved NMRs.
Right. Anything that can be
I'm not sure if we don't have Mesh Nova, so I don't know if we would be able to open that. So it might be better to share the FID file, but we can connect offline about this. That's fine. Sorry, I opened a can of worms.
But it is you know, so some assessment of purity and as much raw data as possible. Is the short.
Okay, and you just want this on in the same drive as the experimental document?
Yeah. Where is that? I'm sorry, I forget. Ed, Daniel, where's the folder for all the experimental
I think it's with
stuff?
the publication.
Is in the same link that you have for the paper, you can link for the file, for the SC file.
Okay. Is it on GitHub?
Yeah,
it's on
yeah.
GitHub. All right. So if it gets too big, then we'll have to think about what to do. But if it can be uploaded there, that would be great. Okay. We are obviously, once we've written this whole thing and got it into a final format, we're going to have to pass it by all the people on the screen. That's going to take a little while. So DNDI will probably have an internal approval process. Wuxi might, I don't know. Also, Laurie, yeah, we'll connect offline about issues to do with the compounds that came through from that route. I know you sent me something about that, and we'll deal with that outside this meeting.
Sounds good.
Okay. Yep. All good.
Okay. Thanks so much, everyone.
Yeah. Thanks very much, everybody. See you again soon.
See you. Bye.
Bye.
Bye.